Hello again. So in this video I'm going to talk about restriction enzymes. Um, in the previous slides, one of the things we talked about is the ability to cut DNA at specific sites so that you are able to join different molecules together. Key word here is specific, right? Now, before we go into that, what are restriction enzymes? These are enzymes that are produced by bacteria for in a defense system, right? You know how viruses, when they infect bacteria, what they do is they insert their DNA in. Now, if the bacteria detects this enzyme, so this foreign DNA, so it will have this enzyme that can actually chop up the DNA and to protect itself. So this scientists were able to isolate this enzyme. This is Werner Aber, Smith and Nathan. They won a Nobel Prize in 1978 for their discovery. And then there are nucleases. Nucleases are enzymes that cut the nucleic acid. It's called nucleases that recognize specific nucleotide sequence in double-stranded DNA and cleave both strands of the duplex. So we're going to look at the process of cutting. This is a typical DNA phosphodiester bond. This is one base and then the other base. So what it does is actually cuts this phosphodiester bond between base 1 and the other base. Alright, okay. So now, this is an example. Look at this side, GAATTC. Okay. Now, how it cuts? It produced two types of or three types of cutting ends. I mean, if you have a string, a piece of a string of DNA, right? If you have two pieces of a string, you can cut them straight, or you can cut one and the other place so that it becomes like that, you know, or it becomes like that. So these are different ways of cutting these two strands of DNA. So GAATTC is a specific site that is recognized by an enzyme called EcoR1. And what it does is that it will cut on the top strand between the G and A. On the bottom strand, it also cuts between G and A. Now look at this sequence. You will find that the top one reads GAATTC. Look at the bottom one, it also reads GAATTC. And they are in complementary. So this G with C, T. So there's nothing wrong with the complementarity. So the top and the sequence are the same, but they are read anti-parallel. And this is what we call as palindromes. And this is a characteristic, one of the important characteristics of restriction sites. These enzymes recognize sites that are palindromic. Another example of palindrome is, let's say, GTAC. Right? If you write down GTAC, the sequence at the bottom, read anti-parallelly, right, will be GTAC as well. So again, it's palindrome. GGCC, the bottom strand is also GGCC, palindrome as well. All right. So these are characteristics of restriction sites. And each enzyme recognizes one specific site. EcoR1, for example, only recognizes GAATTC. It doesn't recognize other sequence. If you want to, we'll look at examples later. But each enzyme will recognize a specific site. Now, in this case, it cuts what you call it does a staggered cut. That means it cuts between G and A at the top strand, and it cuts at GA at the bottom strand. So it gives you, this is what you call sticky. Why is it called sticky? Because remember, DNA molecule likes to be in a double-stranded position. So AATT will attract another complementary sequence AATT and it will bind, right? Okay, so if you cut, let's say this green molecule is the vector, if you cut the vector with EcoR1, right, EcoR1, you cut the fragment that you want to clone also with EcoR1. So this clone also has AATT. This is called protruding end or sticky ends. Now, you'll find that this AATT can pair with that AATT, correct? So it matches, right? And then if you have that, you add ligase, and then it will seal this into a new molecule. Okay? This is in principle. So restriction enzyme, there are many types. You call them type 1, type 2, type 3. Most of the ones that we use in the lab are type 2. They differ in terms of how they cut. Type 1 cut far from recognition site, meaning recognition site is the sequence. So let's say there's an enzyme that recognizes ATAT, for example, just for argument's sake. It recognizes ATAT, but it doesn't cut that. It cuts further away, somewhere downstream or upstream. So that's type 1. Type 2, recognize the site, cuts there. Type 3, recognize the site, but not necessarily cutting it there, but somewhere near. So these are type 3. So in the lab, normally we'll just use type 2 because we know where the site is, it cuts there. Because otherwise, because it needs, sometimes it's difficult to control where it cuts, right? So type 2 restriction enzyme recognizes and breaks double-stranded DNA within particular sequences of tetra, penta, hexa, hepta. This depends on the length of the restriction site. For example, EcoR1 recognizes how many sequences? GAATTC, 6 bases. So this falls under the hexa class, right? Hexanucleotides. 4, 5, 6, 7, 8. And then it's just here, it says, which has an axis of rotational symmetry, the palindromes. Of course, there are exceptions. That means there are sequences that are not palindrome that are also restriction site, but never mind, we will not worry about that. So EcoR1, almost all RE recognizers are palindromes, almost. So it says GATC, EcoR1, reads the same from both sides. These are different types of restriction enzyme. For example, BAMH1, it cuts GGATCC. Again, if you can if you can just do a mental, reading of the complementary sequence, it is GGAATCC. AGATCT complementary is also AGATCT. So these are all palindromes. Now, how do you name the enzymes? Normally, the first letter represents the genus, the next 2 letters represents the species. E. coli is Escherichia, CO. coli. BAM is Bacillus, AM is Amylo lignifications. BGL is Bacillus globii, and so on. PVU is Proteus vulgaris. So, and then the number at the end, R1, H1, it depends on when they were isolated, So, for example, EcoR1, EcoR2, EcoR3, EcoR5, all these have different cutting sites. Sometimes there are also bases in the middle. Hin F1, they cut at GANTC, that means N can be anything. And all this produces sticky ends. Remember, what are sticky ends? They don't cut in a straight fashion. They cut, a staggered. There are examples of blunt end. For example, South3A, okay. LU1 cuts at blunt end. I think that there are slides to show how it cuts to give you a blunt end. Blunt end is also sometimes called flush ends, straight. These are commonly used REs. There are many more. You can Google, especially enzyme companies, and then they will list you all different types of resistant enzymes and what they cut. All right. Okay. For example, NOT1, right? NOT1 is an enzyme that cut the recognition sequence is 8 bases. So GC, GGC, GC. Again, the palindrome is always the same. GC, GGC, GC. This is an example of LU1. Remember, LU1, what is the cutting site? LU1 is AGTC, so AGTC. But for LU1, it cuts straight. So you have a molecule that is AG. It doesn't have any sticky ends. So these are blunt ends, right? But EcoR1 gives you a sticky end, right? Now, look at this. This is another thing you may consider if you're talking about cloning. If you do a lot of cloning, then these are some very useful information. Look at these different enzymes: BAMH1, BAGL1, and SAU3A1. BGL, sometimes we call them BAGL1. So BAMH1, the cutting site is GGATCC. BAGL2, the cutting site is AGATCT. And SAU3A1, the cutting site is GATC. However, for BAM1, the cutting site is between the first and the second G. BAGL2, the cutting site is between A and the first G. And then SAU3A, it cuts at the first G. And you'll find that all three different enzymes give the product of the cut is actually the same sticky ends. It's all GATC. Which means to say, if you cut two different enzymes, one you cut with BAMH1, the other one you cut with BAGL1, they can be joined together because their ends are the same. Alright? So same sticky ends produced by different REs. When you talk about sticky ends, there are two types. Basically, one that says that the overhang is on the 5' molecule. The other one is the overhang is at the 3'. Right? So in this case, EcoR1, it gives you a 5' overhang, or sometimes it's called protruding 5' termini. And an example, PST1, it cuts at G, and then it gives you, this is a little bit off, the drawing is a bit off, right? But basically what it says here is that it has a 3' overhang, or 3' protruding termini. Okay? Okay. So that concludes, well, that's just, we're finishing about resistant enzymes, and in the next video, we're going to look at electrophoresis. All right? I'll see you in the next video.